Hello students, welcome back to your own channel Vedantu NEET English. This is Basparat sir, your biology master teacher. Quickly, let me know in the chat if my voice is audible to all of you, and a beautiful presentation behind me is visible to each and every one of you. Quickly, let me know in the chat. Now, students, this is the part 2 of your photosynthesis in higher plants, part 2. Now, in the first part, we finished all the basics, which includes the experiments, certain types of reactions, what is a pigment, cyclic and non-cyclic photophosphorylation, and that was it. The first part was very solid. We solved a lot of questions, and students who are watching the second part first, I would advise to go first, watch the first part, because that is very important if you want to understand the second part, obviously, right? Obviously. So students, let me know in the chat if I'm audible and visible also. That is very important again. Now, with that being said, what are we going to do today's class? Right? What are we going to do in today's class? The rest of the chapter. That is, we'll be starting off with chemiosmotic hypothesis. It sounds very fancy, but it is very easy to understand. Chemiosmotic hypothesis. Then we'll be doing something called as a dark reaction, C3 cycle. Then we'll be doing C2 cycle. Then we'll be doing C4 cycle. Yes, C3, C2, C4. All in today's class. And we'll be ending the class with something called as factors affecting photosynthesis. Right, that is the end of it. And obviously, towards the end of the class, we'll be solving a lot of questions. That's always there. So I have a lot of PYQs also. So students, stay till the very end. I advise you and I request all of you to stay till the very end to take the advantage, the full advantage of today's session. So with that being said, students, quickly let me know in the chat. Fire emoji in the chat, all of you. Fire emoji in the chat. And we'll be starting off with photosynthesis in higher plants, part 2. Part 2. Okay? Students, quickly mark your attendance, all of you. Quickly mark your attendance, and we will start now. Mark your attendance, all of you. Smash the like button. Now, students, if you are aware, in the previous class we did something called as non-cyclic as well as cyclic photophosphorylation. This is non-cyclic here. Right? This is non-cyclic. We learned non-cyclic. We also learned cyclic. We also learned cyclic photophosphorylation. Yes, I'm Kannadiga. What is that? Yes. Right. Now, students, now we'll be starting off with something called as chemiosmotic hypothesis. Now we'll be starting off with chemiosmotic hypothesis. See, students, it is very, very easy to understand and very simple concept. Very simple concept. Okay. Now, students, if you remember, in the non-cyclic photophosphorylation, quickly tell me in the chat how many ATP and how many NADPH is produced. For everyone, this question is for everyone. Tell me in the non-cyclic photophosphorylation, how many ATP and how many NADPH2 was produced? All of you tell me, sir, there was one ATP and there was one NADPH or NADPH2 was produced in the non-cyclic photophosphorylation. Okay. Under non-cyclic, under cyclic, we got one ATP here and one ATP here. So 2 ATP in the cyclic and 1 ATP in the non-cyclic. 1 ATP in the non-cyclic. Thank you so much for the wishes, all of you. God bless all of you. Right. So that was it. Right. So what does chemiosmotic hypothesis explain then? Chemiosmotic hypothesis explains how this 1 ATP is being produced. It explains how this 1 ATP is being produced right now. So that is what is chemiosmotic hypothesis. Chemiosmotic hypothesis explains how that 1 ATP is being produced, right? How that 1 ATP is being produced. Okay, so that is chemiosmotic hypothesis. But students, I have mentioned earlier, if you go looking around your NCERT, right? If you go looking around your NCERT, you will never find words like pheophytin, plastoquinone, cytochrome B6F complex, plastocyanin, right? Cyanin. If you go look around NCERT, in the any lines of NCERT, you will never find it. You will never find it. Then, sir, where did I bring this all of these equations from, or where does protein complexes come from? Right here, students. If you look at the diagram, of your ATP synthesis, that is chemiosmotic hypothesis diagram here, can you notice here? This right here is nothing but a magnified image. It is a magnified image of a thylakoid. Magnified image of a thylakoid, a single thylakoid. Magnified image of a single thylakoid. And if you look at the membrane of the thylakoid, do you notice it? See here, photosystem 2, plastoquinone, cytochrome B6F complex, plastocyanin, and PS1. Now, quickly tell me in the chat, is this cyclic or non-cyclic? Tell me, is this cyclic or non-cyclic photophosphorylation? So, obviously, if there is photosystem 2 here, then photosystem 1, this right here is non-cyclic photophosphorylation. See, that's right, given right here. Non-cyclic photophosphorylation from photosystem 2 to photosystem 1. And finally, NADP plus, which is the final electron acceptor, getting converted to NADPH2 in the presence of enzyme FNR, ferredoxin NADPH reductase, right? NAD plus reductase. Do you see that here? See, final electron acceptor is NAD plus. It is getting reduced to NADPH in the presence of enzyme FNR enzyme. Given right here. It's given right here. This right here is your non-cyclic photophosphorylation. So the next time someone asks you a question, will they ask you all the protein centers? Yes, they can ask you, because it is actually given your NCERT, but in the form of a diagram. Okay. Now, students, for you to understand now, let's understand chemiosmotic hypothesis. Right. Now, in a chemiosmotic hypothesis, there are four main requirements. Major requirements. The first major requirement, students, is going to be a proton gradient. Or can I say first, let's go from outside. Outside, can we see a first major requirement is going to be a intact membrane. The first one is going to be a intact membrane. Now, after that, a proton gradient. Then a proton pump. Apart from proton pump, there also be requirement of ATP synthase or FOF1 particle. Here, this right here is the FOF1 particle, also called as your ATP synthase. All of you can see here, ATP synthase. Now, students, I want everyone to focus very well now. Super focus. Understand like how each of the component plays a major role. Now, there is intact membrane. Yes, there is intact membrane. There's a proton pump. Yes, there is a proton pump. Yes, there is ATP synthase. As ATP synthase is already present. Now, I want everyone to focus. on the something called as proton gradient. First, understand what is the meaning of gradient, all of you. Madhumita, listen, all of you. What is the meaning of proton gradient? All of you know the meaning of proton is H plus. The meaning of gradient is that a difference in proton, means a difference. So, for a chemiosmotic hypothesis, that is, for a ATP to be produced, I repeat again, for a ATP to be produced, we need a proton gradient, that is a difference in the proton level. Now, between what? There should be a clear difference, students. There should be a clear difference in the proton levels inside the lumen and outside the lumen. For example, inside the lumen, we need high proton. Can you see? High proton. Outside the lumen, we need low proton. Can you see here? Low proton. Now, once this is achieved, high proton on the inside and low proton on the outside. Once that difference is made, ATP can be produced then. ATP can be produced only then. All of you are understanding? Doesn't matter what is mentioned, what is the big deal, learn what is given here. Okay. So, proton gradient needs to be formed. That is, inside the lumen, high H plus, outside the lumen, low H plus. Only when the proton gradient is maintained or achieved, ATP can be produced. Understanding? Now, how do we achieve this proton gradient? How do we make inside the lumen high H plus and outside the lumen low H plus? How do we make it? Now, students, if you remember non-cyclic photophosphorylation, did we learn that there is something called as photolysis of water? Did we learn that? Here. If you go back to your non-cyclic photophosphorylation, did we learn that there is something called as photolysis of water right here? Yes. This photolysis of water which is happening around photosystem 2. Now, what is the byproduct here? The byproduct of your photolysis of water was 2H plus, 1 oxygen and 2 electrons. These 2 electrons were taken up for the reaction. Oxygen was released as a byproduct. And I told you here, this 2H plus is sent for ATP production. 2H plus is sent for ATP production. So that is the first way we are going to make proton gradient. Understand now. Let's come back to this diagram now. If you come back to this diagram, can you see here a green color arrow marks? When there is splitting of water, this H plus is present where? Inside the lumen. It is present inside the lumen. So can I tell more and more, as more and more photolysis of water will happen, more and more H plus will be accumulated inside the lumen. Yes. So more and more photolysis of water is happening here. The inside the lumen H plus is increasing. H plus concentration is increasing inside. All of you will tell me, yes sir, because of splitting of water, H plus concentration is increasing. And it is represented as green color marks here. Yes. Now what about here? Can you see here, students? Plastoquinone. This plastoquinone is also a hydrogen donor. This plastoquinone, with the help of the energy of electron, is able to pull one proton from the stroma. This plastoquinone is able to pull one proton from the stroma into the lumen. Now you tell me, students, everyone in the chat, if a proton is pulled from the stroma into the lumen, will the H plus concentration increase or decrease inside the lumen? All of you tell me now. If the proton is pulled from the stroma into the lumen, obviously the H plus concentration will increase inside the lumen. It will increase inside the lumen. Simultaneously, if we are pulling something from here, so can I tell H plus concentration is decreasing in the stroma now? Obviously, if you are pulling from here, it will decrease here. It will decrease here, right? So by this method, we can establish that yes, inside the lumen, from photolysis of water, splitting of water, and plastoquinone are increasing the H plus concentration inside the lumen. Inside the lumen. Now you tell me, all of you tell me, in the last step of your non-cyclic photophosphorylation, in the last step of non-cyclic photophosphorylation, did we learn that NAD plus gets reduced to NADPH2 by receiving the two electrons, and also receiving 2H plus from the stroma? Did we learn here? That is why, students, I am telling you, everything is connected. Unless you understand this, you cannot understand chemiosmotic hypothesis. All the students are watching it late. This is for you. Okay. So can I tell this particular 2H plus is now again being removed from the stroma? Yes, it is being removed from the stroma. So you tell me in the chat right now, will the H plus concentration decrease or increase in the stroma now? All of you in the chat, all of you. I want everyone to participate. Students, if this particular 2H plus is being removed from the stroma for NAD plus, can I tell again H plus concentration is again decreasing in the stroma. It is decreasing. Yes, decreasing in the stroma. Understood? So can I say that now there is an establishment of a proton gradient? There's a difference in proton level now. I can confidently say that, yes sir, inside the lumen there is a high proton and outside the lumen, that is in the stroma, there is low proton gradient now, low protons. So with the help of that, we have established a proton gradient now. Now once the proton gradient has been established, it is made ready. 4H plus, students, 4H plus, when 4H plus are sent into the ATP synthase, when 4H plus are sent into the ATP synthase, it can convert 1 ADP into 1 ATP. And that is how we get ATP molecules as the product of your light reaction. Understood, all of you? So that's why we say, right, in non-cyclic, ATP is made here. Every single time there is one splitting of water, there is one ATP is always made. Understood, all of you? So chemiosmotic hypothesis explains how the ATP is made, and you just now learned how it is made. Quickly tell me in the chat any doubt here. No doubt? All of you? No doubt? All of you understood? Chemiosmotic hypothesis, very simple to understand. The chemiosmotic hypothesis requires 4 main things that is given here: intact membrane, proton gradient, proton pump, and ATP synthase. And this is how there are 3 ways in which a proton gradient can be established. The first method is splitting of water. Second method is plastoquinone. Third method is going to be the final ADP. NADP gets converted to NAD plus. It is consuming a proton from the stroma. That's all. 3 methods to establish the proton gradient. Now let's read what is given in NCERT. All of you read with me now. Okay. Let us now try and understand how actually ATP is synthesized in the chloroplast. See, I told you, chemiosmotic hypothesis explains how the ATP is being produced. The chemiosmotic hypothesis has been put forward to explain the mechanism. Like in respiration, in photosynthesis too, ATP synthesis is linked to development of proton gradient. I told you, we just now learned. If you want photosynthesis to happen, if you want ATP formation to happen, we need proton gradient. Across the membrane. This time, these are membranes of thylakoid. See, it happens on the membrane of the thylakoid. There is no difference though. Here, the proton accumulates towards the inside of the membrane, that is in the lumen. We learned that. We learned just now that inside lumen, there is increase in what? Increase in H plus concentration inside the lumen. We learned just now, right? Okay, inside the lumen, right? Now, in respiration, proton accumulates in the intermembrane space of the mitochondria. This you will learn in the electron transport system, next chapter. Okay. When electrons move across the ETC, let us understand what causes the proton gradient across the membrane. We need to consider again the process that takes place during the activation of electrons and transport to determine the steps that are cause the proton gradient to develop. So basically telling you here in this paragraph is that if you want to understand chemiosmotic hypothesis, you need to understand non-cyclic and cyclic photophosphorylation. That's all in this paragraph. See here, there are 3 different ways. What is the first one, students? Since splitting of water molecule takes place on the inner side of the membrane, the proton or hydrogen ions that are produced by the splitting of water accumulate within the lumen of the thylakoid. The first method I told you, which is accumulation of proton with the help of splitting of water inside the lumen. Okay. Now, second one, as electrons move through the photosystems, protons are transported across the membrane. Yes, protons are transported across the membrane. This happens because the primary electron acceptor, tell me students, which is the primary electron acceptor in the case of non-cyclic photophosphorylation? All of you in the chat, tell me. Which is the primary electron acceptor in the case of non-cyclic photophosphorylation? The primary electron acceptor is pheophytin. All of you remember. All of you remember the primary electron acceptor non-cyclic is going to be pheophytin, right? Pheophytin, right? Pheophytin. Now this happens because the primary acceptor of electrons, which is located towards the outside of the membrane, transfers its electron not only to an electron carrier, I told you, PQ, CY, PC, electron carriers, but to an hydrogen carrier. Okay, hence this molecule removes a proton. I told you, which is this? This molecule we are talking about. This molecule they are talking about is none other than plastoquinone. This plastoquinone is also a hydrogen donor. It is also a hydrogen donor right here. Molecule removes a proton from the stroma while transporting an electron. So when it is transporting an electron, it is also pulling a proton from the stroma, right? When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side of the lumen side of the membrane. So I told you here. The second one is what? Plastoquinone is pulling a proton and it is sending inside the lumen. So whatever we have thought is already given in NCERT, right? Now, third one. The NAD plus reductase enzyme. The last one here. NAD plus reductase enzyme, FNR enzyme. Okay, FNR enzyme. Okay. The NAD plus reductase enzyme is located on the stroma side of the membrane. You see here, all of you. This NADPS reductase right here, students, is located on the, can you see here? This is the stroma. This is the stroma side. Located on the stroma side, FNR. Stroma side, right? Stroma side. Right? Located on the stroma side of the membrane along the electron that comes from the acceptor of the electron of PS1, right? Electrons are coming from PS1 now, right? Electrons are coming from PS1. To this NAD plus, electrons are coming from PS1. Everything is given here. Protons are necessary for the reduction of NAD plus to NADPH2. That's why I wrote NADPH2. See, NADPH plus H plus. NADPH2. NAD plus is getting reduced to NADPH2. These protons are also removed from the stroma. So when there is reduction is happening, one particular proton is removed from the stroma. So can I say there's a decrease in the H plus level in the stroma now? Yes, given here. Hence, within the chloroplast, protons in the stroma decrease in number, while in the lumen there is an accumulation of protons. So inside the lumen, proton is increasing. In the stroma, it is decreasing. That's all. This creates a proton gradient across the thylakoid membrane as well as the measurable decrease in pH in the lumen. Obviously, more H plus inside the lumen, decrease in the pH, obviously. Now, why are we so interested in the proton gradient? Now, this gradient is important because it is the breakdown of the gradient that leads to the synthesis of ATP. Now, once this gradient is produced, students, once this gradient is made, this balance is made. If you break the gradient, what happens? If you want to break the gradient, what do? Tell me, all of you now. Now there is inside the lumen, there is more H plus, outside less H plus. Now if you want to break down the gradient, obviously you have to push the H plus outside now. If you want to make it balanced again, this H plus has to be pushed outside, obviously. So only when the H plus are pushed outside, the gradient is again back to normal. Yes, it will be back to normal then. Yes. Now understand what is given here. Why are we so interested in the proton gradient? This gradient is important because it is the breakdown of this gradient. That is, we are breaking the gradient now. Now gradient is there now. Now inside the lumen there is very high H plus, outside very low H plus. Imagine if you have to make it level. What do we have to do? You have to push the electrons, we have to push the protons outside the lumen. Only when we push the protons outside the lumen, the gradient will be again back to normal. Yes. So that is called as the breakdown of the gradient. That leads to the synthesis of ATP. The gradient is broken down due to the movement of protons across the membrane to the stroma. I told you, they will be pushed outside the stroma again. They will push into stroma. We did that here. All the protons are being pushed into the stroma again. Why? To break the gradient. To break the gradient. They are pushed outside the stroma through the transmembrane channel of CF0 of ATP synthase. So when the protons are being pushed outside, they are being pushed outside through the ATP synthase. Right? The ATP synthase enzyme consists of two parts. One is called as F0. The above part is embedded in the thylakoid membrane. Can you see? Students, the above one is called as the F0 here. This region, the first one is called as the F0. This is the F0, which is inside the membrane. This is the F1 particle. This is the F1. This is F0. F0, F1. Okay. Is embedded in the thylakoid membrane and forms a transmembrane channel that carries out the facilitated diffusion of the protons across the membrane. The other portion is called as the F1 and the protrudes out of the outer surface of the thylakoid. See, F1 is outside. F1 is outside. Membrane on the side of the face of the stroma. So F1 is towards the stroma. F1 is towards the stroma. See, F1 is towards the stroma. This is stroma, right? This is the stroma. This is the stroma. Stroma. Right? Stroma. The breakdown of the gradient provides enough energy to cause the conformational change in the F1 particles of the ATP synthesis, which makes the enzyme synthesis several molecules of energy of packed ATP. So I told you, every single time 4H plus is pushed outside, 1 ATP is made. 1 ATP is made. Chemiosmotic hypothesis requires a membrane. I told you, it requires the intact membrane, a proton pump, a proton gradient, and ATP synthase. The energy is used to pump the protons across the membrane, creates a gradient or high concentration of protons within the thylakoid membrane. Right, that is the function of proton pump. ATP synthase has the channel that allows the diffusion of the protons back into the membrane. That is, they are again coming back into the membrane. This releases enough energy to make the ATP. Along with NADPH, that is the complete result of non-cyclic. NADPH produced by the movement of electrons, the ATP will be used immediately in the biosynthetic reaction. Students, can anyone tell me what is this biosynthetic reaction? What is this biosynthetic reaction, students? Where have you heard it? This biosynthetic reaction is nothing but the dark reaction. So we learned it before. The products of your light reaction is used in the dark reaction. The products such as your ATP and NADPH is used by the dark reaction. Like how your mother makes the food, you only eat the food, right? Your mother is making the food, you are eating the food. Same thing is happening now. Light reaction is making the food, dark reaction is eating the food. But dark reaction is eating the products, but it is also making glucose. You are not making any glucose. Okay. Taking place in the stroma, responsible for the CO2 and synthesis of sugar. That is dark reaction ending. Friends, any doubt you quickly ask me, students. That was the complete, functioning of your entire chemiosmotic hypothesis. Yeah, you're only eating, not making. Only thing you'll make is Maggie. On 2 minutes, Maggie will make off. Any doubt here? Very simple, no? Very simple it is. This was a complete chemiosmotic hypothesis. How ATP is made. Okay. Any doubt there? Any doubt? No doubt, right? Very easy. Now, students, similarly, the entire chapter is going to be, rest of the part is going to be very simple, okay? You don't have to worry about it. Now, we'll be doing something called as where ATP and NADP is used. Like, what is the next step? What is the next step of it, right? After that, we will learn something called as C3 cycle. Then we learn something called as C2 cycle. Then we will learn something called as your C4 cycle. Everything is very easy. And let me tell you, students, whatever you learn right now, because this is going to be a very long lecture, right? Whatever you learn right now is the foundation, the basics which you're learning. There is so much more, okay? And since you are sitting here for hours together, and I know you will, okay? Trust me, whatever you study right now will be useful to you, and you can check on your own. How do you check? After the class, since anyway you're reading NCERT, try solving questions, as many questions as possible, and tell me even if you one question you cannot do. If the question is from NCERT, you will be able to do every single question from this chapter. That is what I do. So when I'm teaching, I make sure that you get to know every single. So where are you from? I'm from Bangalore. Vampire, huh? A nice name. What? No, vampire. Okay. Now let's start with where do ATP and NADPH is used. Students, can anyone tell me what is the answer to this? What is the answer to this? I've been telling you from that time. Okay. What is the answer to this question? Where are the ATP and NADPH are used? Where do you think they'll go? Obviously, don't tell your stomach. Obviously, they'll go to your stomach only. But in the case of plants, where are they going? In dark reaction. Exactly. Now they're going to dark reaction now. Now tell me, does the dark reaction happen in darkness? Obviously not. That does not happen in the darkness. Okay, they don't happen in the darkness. Now, we learned that products of a light reaction, ATP and NADPH and oxygen, three of the important products of these O2 diffuses out of the chloroplast, right? It goes outside the chloroplast because it is not required. Like, imagine when you're eating biryani, do you eat the elaichi? Do you eat the spices? No. You throw the spices? No. Like once it is used, you throw it out. So here also, they don't require oxygen. So oxygen is thrown out. Like how in biryani, if you eat that clove and everything, you throw out. No. Here also, oxygen is being shoot out once after use is over. Okay. Diffuse out of the chloroplast. While ATP and NADPH are used to drive the process of leading to the synthesis of food. More accurately, sugar. Okay. This is the biosynthetic phase of photosynthesis. This process does not directly depend on the presence of light, right? So can we call them light independent? Yes. Can we call them light independent now? They don't require light, right? All the work is done by your light reaction. No importance given to light reaction only after that. All that. Okay. No importance given to light reaction. Sad. Okay. But it is dependent on the product of a light reaction, ATP and NADPH, besides CO2 and H2O. You may wonder how this could be verified. It is simple. Immediately after the light becomes unavailable, the biosynthetic process continues for some time and then stops. If then light is made available, the synthesis starts again, right? Synthesis starts again. So students, tell me, does the light reaction happen in the daytime? Tell me, students. Does the light reaction happen in the daytime? Yes, sir. Does the dark reaction happen in the daytime? Yes. Dark reaction also happens in the daytime. Also happens in the daytime. Okay. Can we hence say that calling the biosynthetic phase as a dark reaction is a misnomer? Misnomer basically means you think dark reaction happens in darkness. It does not happen like that. That's what they're saying. Okay. No. Friends, can we start dark reaction now? Before we read any further, let's quickly start the dark reaction. Very simple dark reaction. I will draw it for you. Right, I'll draw for you and draw with me and understand. Write down. I will tell you exactly from where the questions will come. Apart from that, they will not ask anywhere. They'll not ask anywhere. Okay, ready? First, we'll do dark reaction now. Dark reaction. Dark reaction. Understand? Okay. Understand dark reaction. Now this dark reaction, students, this dark reaction was found by a scientist, Melvin Calvin. Okay, not Calvin, not Calvin milkshake. First I thought it was like Calvin milkshake only, but it is not. But Calvin. Okay, Melvin Calvin. So it is also called as Calvin cycle. We can also call it Calvin cycle. Okay, Calvin cycle. Very simple. Calvin cycle or dark reaction. Okay, or dark reaction. So this entire reaction, students, this entire reaction starts with something called as RUBP. It starts with RUBP. RUBP. This RUBP is nothing but rubulose 1,5 rubulose 1,5 bisphosphate. Rubulose 1,5 bisphosphate. Okay, or biphosphate. You can call it biphosphate or bisphosphate. Okay. Now this RUBP is a 5-carbon compound. Okay. Friends, to this RUBP, one CO2 is entering the party. All of you know, right? All of you know plants require CO2. All of you have already learnt plants convert CO2 into C6H12O6. Yes, they convert CO2 into C6H12O6. Yes. Now, so this RUBP is taking one carbon. How many carbons here? One carbon. Now, students, tell me, what is 5 plus 1? What is 5 plus 1? You will tell me, sir, 5 plus 1 is 6. So, technically, aren't we supposed to get a 6 carbon compound, students? Aren't we supposed to get a 6 carbon compound here as a product? 5 plus 1 is 6, all of you know. So, shouldn't we get a 6 carbon compound? So, all the students, all the, not only you, even the scientists were also looking for a 6 carbon compound. But they never got. A 6 carbon compound. In fact, they got a 3 carbon compound. All of you. 3 carbon compound. Okay. That is called as PGA. 2 molecules of 3 PGA. What is PGA? PGA is nothing but phosphoglycerate. 3 PGA. Okay, 3 PGA. And students, remember, this entire process of conversion of RUBP into 3 phosphoglycerate or 3 phosphoglycerate is enzyme mediated. It is completely enzyme mediated, and the enzyme involved here is something called as Rubisco enzyme. Rubisco enzyme is the enzyme which is making sure that RUBP gets converted to 2 molecules of 3 carbon PGA. And this first step is called as carboxylation. This first step right here is called as carboxylation. First step is called as carboxylation. RUBP to 2 molecules of PGA. So 3 carbon, 3 into 2 is 6 carbon. Right? 6 carbon. Now students, understand. Now this phosphoglycerate gets converted to another 3 carbon compound called as triose phosphate. And this triose phosphate, students, this triose phosphate eventually gets converted to glucose. Which all of you need to eat food. Or plants require their own food. This is the thing. That is glucose. This is the glucose. Okay, it's converted to glucose. But in the second step, it is not very easy. It comes at a cost. The second step is not very free. It actually comes with a cost. What is the cost? The cost here is 2 ATP and 2 NADH. The cost here is getting converted to ADP. 1 NADPH is getting converted to two of them, NAD plus. NAD plus. How many of them? Two of them, not one. Two of them. Two ATP and 2 NADPH is being consumed here. They being consumed. Okay. Clear? Clear all of you? Clear? Not any doubt? And this second step, students, this second step here, the second step in which there is something called as can you tell us something is reducing here? We are losing so many things, right? We are losing NADPH, we are losing ATP. So can I call it reduction step? Reduction. Reduction. Second step is called as reduction. Now, this triose phosphate again gets converted back to the RUBP. It again gets converted back to the RUBP. Last step. Now, in the last step also, it is not very free. Last step is also not free. It comes at a cost again. The cost here is very simple. 1 ADP. 1 ADP gets converted to 1 ATP. Sorry. Ulta. 1 ATP gets used to 1 ADP. It is losing again. 1 ATP is again consumed. 1 ATP is again consumed. Now, students, can I tell again, since we are getting RUBP again, so can I call it regeneration now? Can I call this step regeneration? Regeneration. This is the entire cycle of dark reaction. Now, students, tell me, in this particular cycle, which is the first stable product? Students, which is the first stable product here? Obviously, it was here. The first stable product was PGA. How many carbon? 3 carbon. So, can I call this C3 cycle? C3 cycle? Yes, because 3 carbon is the first stable product here. So, can I call it C3 cycle? That's all. That's all is the dark cycle, Calvin cycle, also called as C3 cycle. C3 cycle. Okay. Now, students, tell me, for one CO2 right now, for one CO2, how many ATP was consumed? Tell me. For every one CO2, students, for every one CO2, can I tell 2 ATP there, 1 ATP here, 3 ATP was consumed, and 2 NADP was consumed. 2 NADPH was consumed for every one CO2. Now you tell me, this is for one CO2. How many will be required for a glucose? How many ATP and NADPH will be used for a one glucose? For one glucose. Understand here. Since CO2 is only 1 carbon, glucose is nothing but C6H12O6. 6 carbon. For 1 carbon it is 3 and 2. For 6 carbon, how much will it be? For 6 carbon, just multiply into 6. That's all. 3 ATP into 6. 2 NADPH into 6. What do you get? 18 ATP, 12 NADPH. 12 NADPH. So for one glucose, there will be a requirement of, students, there will be a requirement of 18 ATP and 12 NADPH. 18 ATP and 12 NADPH. Don't get confused, this ATP is from, this ATP and NADPH is from the cycle, okay, don't get confused. 12 NADPH. That's all is C3 cycle, done. That's all is the C3 cycle. Any doubt, you quickly ask me, any doubt here? It is very fairly simple. Carboxylation, reduction, regeneration. RUBP to 3-PGA. Then we have triose phosphate, which can convert to glucose. Triose phosphate is getting converted back to RUBP. Rubulose 1,5 bisphosphate. Done. This is called as dark reaction. This is called as dark reaction. Now let's see what is given in NCERT. Let's see it, all of you. All of you read with me now. All of you should read with me so you can understand better. Let us now see how the ADP and NADPH are used in the biosynthetic phase. We saw earlier that CO2 is consumed with H2O to produce the glucose or sugar. It was interest to scientists to find out how this reaction proceeded, or rather what was the first stable product of when CO2 is taken into the reaction or fixed. Just after World War II, among the several efforts were to put radioisotopes to beneficial use, the work of Melvin Kelvin is exemplary. The use of radioactive 14C by him in algal photosynthesis studies led to the discovery that the first CO2 fixation product was the 3-carbon organic acid. That is nothing but your PGA. Okay, PGA. He also contributed to working out the complete biosynthetic path. Hence, it is called as Kelvin cycle. That is why it's called as Kelvin cycle after him. The first stable product is 3-phosphoglyceric acid or 3-phosphoglycerate. Same thing, glycerate. In short, PGA. Okay. How many carbon it has? It has 3 carbon. It has 3 carbons. Scientists are also trying to know whether all the plants have PGA in as the first product of fixation, or whether or any other product that was formed in the other plants. Experiments conducted over a wide range of plants led to the discovery of another group of plants where the first stable product of the CO2 fixation was again an organic acid, but one which has 4 atoms in it. This acid was identified as oxaloacetic acid, which we will learn later on. This we will learn in C4 cycle. That is OAA. OAA, oxaloacetic acid. Oxaloacetic acid, also called as OAA. Since then, CO2 assimilation during photosynthesis was led to be of 2 main types. The plant which is the first stable product of CO2 fixation is C3 is called as C3 pathway or C3 plants, or in certain plants where the first stable product was a 4-carbon compound are called as C4 plants. Okay, and those in which the first product was a C4 acid, they are called as C4 pathway. The two groups of plants showed other associated characters as we discuss later. Okay, primary, this is what we learned right now, students. Primary acceptor of CO2. Tell me, students, which is the primary acceptor of CO2? This is a simple question, all of you. Which is the primary acceptor of CO2 in your C3 cycle? We just now learned. Quickly go. Let's see how many can answer. Let's see who can answer first. The primary acceptor of CO2. Primary acceptor of CO2 in your C3 cycle. Meghana, exactly. PGA. PGA is the product. PGA is the product of your first product. First acceptor of CO2. PGA is the product. No. RUBP. Exactly. Ravindrani, it was RUBP. The first acceptor of your CO2 is RUBP. The first product is your PGA. Understood the difference, students? Understand the difference? Exactly, Surya. Exactly. Vinita, you should know it is a product is PGA. The first acceptor is going to be RUBP. RUBP. First acceptor is RUBP. The first product is going to be PGA. Right? See, let us now ask ourselves a question that was asked by scientists who were struggling to understand dark reaction. How many carbon atoms would a molecule have after accepting CO2? Would have 3 atoms of PGA. The study very unexpectedly showed that acceptor molecule was a 5-carbon keto sugar. A 5-carbon keto sugar called as ribulose 1,5 bisphosphate. Now you'll be wondering, so 5 plus 1 is supposed to be 6. They were thinking that only. Do not worry, the scientists also took a long time to conduct many experiments to search this conclusion. They also believe that since the first product was a C3 acid, the primary acceptor would be a 2-carbon compound. Why 2-carbon compound? Because 2-carbon compound plus 1 CO2 should give us 3-carbon compound, that is PGA. So they were thinking the first acceptor is going to be a 2-carbon compound. Okay, understood? They were thinking the first acceptor was going to be a 2-carbon compound. So if the first acceptor is a 2-carbon compound plus CO2, we will get 3 PGA. It makes sense now. It makes sense. It makes sense. But they could never find. They spent many years trying to identify the 2-carbon compound. They could never find this 2-carbon compound. Why? Because it was never there. It was never there. What was there? A 5-carbon RUBP. So what did they find? They found a 5-carbon RUBP plus this 1 CO2 will give you 2 molecules of 3 PGA. This is the right one now. This is the right one. Understood? Surya Prakash. They were trying to identify a 2-carbon compound. Why? Because they thought if there is 2 carbon here, 1 carbon is from CO2, we will easily get a 3 carbon. 2 plus 1 is 3 PGA. So scientists were looking for a 2-carbon compound, but they could never find it all. They're like, where is this 2-carbon compound? Where is it hiding? They could never find. But what they actually found later was a 5-carbon compound. Why? 5 plus 1 is 6, right? 6. So what did they do? 2 molecules of 3 PGA. So 3 carbon into 2 is 6 carbons. Now it makes sense, right? Now it makes sense. 5 plus 1 is 6. 3 into 2 is also 6. 6 carbon here, 6 carbon here. That's what was the conclusion out of it. Understood? They could never find this. It was never there. Now, Kelvin cycle. The Kelvin and his co-workers then worked out the whole pathway and showed the pathway operated by Kelvin cyclic manner, the RUBP. The RUBP was generated. Let us now see how the Kelvin pathway operates and where the sugar is synthesized. Let us at the outset understand very clearly that the Kelvin pathway occurs in all photosynthetic plants. It does not matter whether it is C3 or C4. Very important line. Very important line. Very important. Understand this line. Understand this line. Very important line. The most important line here for you to understand here is C3 cycle will always happen in all the plants which has chlorophyll A. All the plants. It will happen in every single plant. C3 cycle. It doesn't matter if it's a C4 plant. You will understand why later on. It doesn't matter if a plant is C4 or C3. C3 cycle will happen even in C4 plants. Even in C4 plants. I'll prove you that in the next few minutes. For ease of understanding, the Kelvin cycle can also be described under the 3 stages: carboxylation, reduction, and regeneration. 3 different steps. First step, carboxylation. Carboxylation is the fixation of CO2 into the stable organic intermediate. Carboxylation is the most crucial step of the Kelvin cycle, where CO2 is utilized for a carboxylation of RUBP. So RUBP is attaching with CO2. This reaction is catalyzed by an enzyme, RUBP carboxylase oxygenase, Rubisco, which results in the formation of 2 molecules of PGA. Since this enzyme also has oxygenase activity, it would be more correct to call it RUBP carboxylase oxygenase or Rubisco. What is CO? CO here is nothing but carboxylase and oxygenase. Did you understand that, students? Here the first step. This is the first step we are talking about here. RUBP carboxylation, Rubisco enzyme, 2 molecules of 3 PGA. This is the first step. Second step, what is second step? Reduction. These are a series of reactions that led to the formation of glucose. See, obviously, in the second step, glucose is being produced. All of you agree with me? In the second step, glucose is being produced here. If you look here, students, in the second step, that is reduction phase, glucose is being produced here. Glucose is being produced. Second step. The step involves utilization of two molecules of ATP for the phosphorylation, two of NADPH for reduction for CO2. So in the reduction step, there is usage of 2 ATP, 2 NADPH molecule. The fixation of 6 molecules of CO2 and 6 turns of cycles are required for 1 molecule of glucose. Why? Because glucose is always C6H12O6. For 1 CO2 fixation, 1 time the cycle. 6 time fixation, 6 carbon fixation, 6 times the cycle has to turn. 6 times. Now last step is regeneration. Regeneration of CO2 acceptor molecule RUBP is crucial if the cycle is to continue. Obviously, without Rubisco, the entire cycle cannot happen, right? And the regeneration step requires 1 ATP for the phosphorylation to form RUBP. So in the last step also there is usage of 1 ATP. 1 ATP is called as regeneration. Triose phosphate again back to Rubisco. That's all. That's all is the entire thing happening. Clear, all of you? See here. Rubisco, carboxylation, CO2 is coming. Phosphoglycerate or phosphoglyceric acid. ATP, 2 ATP, 2 NADPH. Reduction. Triose phosphate, glucose starch. Regeneration, 1 ATP is used. Again, Rubisco. Again, RUBP. Again, back to RUBP. Students, with that, your C3 cycle is completed. Tell me, is C3 cycle difficult? Tell me, is C3 cycle difficult at all? Obviously not. C3 cycle is super easy. The only thing they'll ask you question is right here. I have already told you this. For 1 glucose, 18 ADP is used of, 12 NADPH is used of, 12 NADPH, right? 18 ATP and 12 NADPH. 12 NADPH. Done. Same thing they'll repeat again. Over the years, the same question is always asked. Okay. Now, students, you tell me. Now, for example, now let's take I am Rubisco. Tell me, what are my two properties? Tell me, students, what are my two properties? Either I can bind with oxygen in my right, students. If I am Rubisco, in my right side I have oxygen, in my left side I have CO2. Okay, now what can happen here? Understand. So I am Rubisco. I can always form a bond with CO2. So if I form a bond with CO2, usually C3 cycle takes place. So I am Rubisco. I am forming a bond with CO2 and C3 cycle happens. But is Rubisco, students, Rubisco CO means what? Carboxylase and oxygenase. Can I form a bond with oxygen too? Tell me, can I form a bond with oxygen now? So when a Rubisco, students, when a Rubisco forms a bond with oxygen, that leads to C2 cycle. So Rubisco can form a bond. Rubisco is very greedy. Rubisco can form a bond with CO2. It can form a bond with oxygen. When it forms a bond with CO2, C3 cycle happens. When it forms a bond with your oxygen, C2 cycle takes place. And the other name for a C2 cycle is called as, can anyone tell me in the chat? What is the other name of a C2 cycle, which is also referred to as a wasteful process? It's a waste body. Tells you, mother tells you, no. When you're not doing any work in the house, what do they call you? Waste body. Even they call me also waste body in the house sometimes when I don't do work. But this right here is the waste body cycle. Waste cycle. What is the cycle called as? Waste cycle. There is no use to plants from this cycle. Anyone? What is the cycle called as? It is called as photorespiration. It is called as photorespiration, also called as C2 cycle. C2 cycle. C2 cycle. Photorespiration. Now, students, came and tell me in the chat. Can we start photorespiration now? I will teach you photorespiration in just 2 minutes. Ready? I'll teach you photorespiration in just 2 minutes. Just 2 minutes. Can we start photorespiration in just 2 minutes? It's very simple concept, very small concept, very small concept. In just 2 minutes, we can do it. Just 2 minutes. Okay. Now, the primary acceptor of oxygen. The primary acceptor of oxygen in the case of C2 cycle is also RUBP. It is also RUBP. Again, RUBP. RUBP. Plus oxygen. Plus oxygen. Now tell me, students, RUBP was 5 carbon. Yes, it was 5 carbon. Oxygen, 0 carbon. 0 carbon. So tell me, technically, you are supposed to get a product of your 5 carbon product. Yes. Now, here what happens is, instead of getting a 5 carbon, we are getting a 2 carbon. We are getting something called as PG, which is 2 carbon plus PGA, phosphoglyceric acid or phosphoglycerate, same as before, is 3 carbon. 3 carbon. So 5 carbon, 0 carbon. Here we have 2 carbon and we here have 3 carbon. Now you will ask me, sir, what is this PG then? What is this PG? The PG here is nothing but phosphoglycolate. Phosphoglycolate. What is PGA? Phosphoglycerate. Similar, same thing as before. Phosphoglycerate or phosphoglyceric acid. Okay. Now this phosphoglycerate, glycolate will again somehow end up getting converted to PGA eventually. Eventually it will become PGA. And this entire reaction is enzyme mediated by none other than Rubisco enzyme. Now in this entire reaction, students, there is no sugar formation. No sugar formation. There is no ATP formation. But ATP is being consumed here. ATP is being consumed here. Is it taking oxygen? No, it is not taking oxygen. It is not taking CO2. It is not taking CO2. So can I tell CO2 is being escaped out? Yes. Since it is not using CO2, CO2 is being escaped out. That's why, students, that's why this is called as photorespiration. Photorespiration or C2 cycle. Why C2 cycle? Because the first stable product is nothing but a 2 carbon compound. That's why C2 cycle. Before it was called a C3. Why? Because PGA was 3 carbon, C3 cycle. Here we have 2 carbon, so C2 cycle, and that's it. Done. That's all. It's C2 cycle. That's all is your entire C2 cycle. Just see here what is given. Let us try and understand how one more process that creates an important difference between C3 and C4 plants is photorespiration. So photorespiration creates a difference between C3 and C4. Why? Why is it creating a difference? Because, students, this C2 cycle right here will never happen. Very important point. Write down. This C2 cycle right here will never happen C2 cycle will never happen in C4 plants. In C4 plants. Never happen in C4 plants. The most important point here. This C2 cycle will never happen in C4 plant. This C2 cycle will always happen in C3 plants only. C3 plants only. So can I tell C4 is a little bit advanced now? C4 is a little advanced? Yes, it is advanced. Okay. C4 plants are very smart plants. C4 plants are very smart plants, that's why. Smart plants. Okay. That's what is given here. To understand photorespiration, we have to know a little more about the first step in the Calvin cycle. The first CO2 fixation step. This is the reaction where RUBP combines with CO2 to form a 2 molecule of 3-PGA. Yes, 2 molecule of 3-PGA in the case of Calvin cycle. Enzyme is Rubisco enzyme. This is what happens in the carboxylation step. Yes, carboxylation, this happens. Rubisco, that is the most abundant enzyme. Very important line. Rubisco is the most abundant enzyme, PYQ, in enzyme in the world. Do you wonder why? It is characterized by a fact that its active site can be bind to both CO2 and oxygen. I told you, it can bind to CO2 and also oxygen. Both they can form a bond with oxygen, form a bond with CO2. Very smart, very greedy, Rubisco. Okay. Now can you think of how this could be possible? Rubisco has such greater affinity for much greater affinity for CO2. Yes, affinity for CO2. When CO2 is to oxygen ratio is nearly equal. When both are present equally, it will go for CO2 only. Imagine what would happen if we're not so. That is, what would happen? What would happen, students? What would happen if the oxygen concentration would go high? If the oxygen concentration would go very high, then obviously then CO2 cycle will happen. CO2 cycle will happen. Or it can happen when the CO2 is very low. So usually when they are equal, when CO2 and oxygen both are equal, it will always go for CO2. Rubisco will always go for CO2 when they are equal. But as soon as oxygen goes high, it forms a bond with Rubisco. On backstabbing friend, this is backstabbing friend. Okay. Now what about whenever CO2 also goes down, it is leaving the side of CO2, it is a forming bond with oxygen, Rubisco. So when will C2 cycle occur? C2 cycle will occur when oxygen is very high or CO2 is very low. Or can I also say very high temperature, during high temperature and also during high light conditions. In these 4 different places, C2 cycle will occur. Okay. C2 will occur. Because it has because your Rubisco, the active site it can bind really well with CO2. The configuration is much more. Imagine when proper key is there, lock is there. It can easily fit there to the active site. Okay. That's why when they're in equal, locking key very easily. When oxygen and carbon dioxide are present together, CO2 binds very well with the Rubisco. Okay, because of the configuration. See here, not so. This would be competitive, right? Then it becomes competitive. It is relative concentration of O2 and CO2 that determines which two will bind to the enzyme. In C3 plants, O2 does bind to Rubisco and hence CO2 fixation is decreased. So in C3 plants, RUBP is binding with oxygen. Here RUBP, instead of being converted to 2 molecules of PGA, binds with oxygen to form 1 molecule of phosphoglycerate and phosphoglycolate. 1 molecule of phosphoglycolate in a pathway called as photorespiration. Did we just learn that here? 1 molecule of Pg, 1 molecule of Pg and 1 molecule of PGA. 1 here, 1 molecule of phosphoglycerate and glycolate. Okay, 2 carbon in a pathway called photorespiration. In the photorespiration pathway, there is neither synthesis of sugar nor ATP. Nothing is released. Rather, it results in the release of CO2, which is utilized for ATP. In the photorespiration pathway, there is no synthesis of ATP or NADPH. The biological function of photorespiration is not known here. That's why we call it a wasteful process altogether. And that's C2. Okay. In C4 plants, photorespiration does not occur. Does not occur. Does not occur. Very beautifully said. This is because they have a mechanism that increases the concentration of CO2 at the enzyme site. So what are they doing, students? What they are doing is very simple. Now imagine, let's go back here. Let's go back here. Imagine this is normal C3 plants. Now what is C4 plants doing? C4 plants are doing very simple thing. What they are doing is, in C4 plants, now we'll take one C4 plant here, C4 cycle separately. In C4 cycle, they're making a separate room. They're making a separate room in which they have the CO2. In which they have CO2. And in this room, they're also locking the Rubisco. So what they're doing is, in C4 cycle, they're making a room in which Rubisco and CO2 is completely locked inside. And oxygen cannot enter this room. Oxygen cannot enter this room at all. So tell me, students, in the case of C4 plants, is it possible to have photorespiration then? Is it possible that this Rubisco right here, can it bind with oxygen? No. This Rubisco right here cannot bind with oxygen. Why? Because Rubisco and CO2 are locked in a separate room or separate chamber. They're locked and kept. Where oxygen cannot enter there. If oxygen is not entering, there is never photorespiration in C4 plants. Never photorespiration in C4 plants. Understood, all of you? Tell me, sir. Understood photorespiration? What happens in your C4 plants? Okay. This is because they have a mechanism that increases the concentration of O2 at the enzyme site. So in the enzyme site, there will be only and only CO2, no oxygen. Okay. This takes place when the C4 acid from the mesophyll is broken down in the bundle sheet to release CO2. We will learn this. We will learn this. We will learn this. We will come back to this slide. We will come back to this. If I teach you right now, you will not understand this. Let's read it right now. I will teach you this again. This takes place when the C4 acid from the mesophyll is broken down. C4 acid, I'll tell you the acid, malic acid, is broken down in the mesophyll cell. There are two types of cells. Okay, mesophyll and bundle sheet. Is broken down in the bundle sheet to release CO2. This results in the increase in the interaction of CO2 in the concentration of CO2. In turn, ensures that the Rubisco functions as a carboxylation, minimizing the oxygen's activity. So basically, in the bundle sheet, there will be no CO2. We'll come back here. Okay. Now that you know that C4 plants lack photorespiration, you probably can understand why productivity and yield are better in these plants. So productivity and yield is always better in the case of your C4 plants. In addition, these plants show tolerance to high temperature, very high temperature can tolerate. Based on the above discussion, can you compare the show? We will learn this between the C3 and C4. Comparison we'll do later on. Now let's understand C4 pathway. Let's read this line. Plants that are adapted to dry tropical regions. What is the time now? Okay. Plants that are adapted to dry tropical regions have C4 pathway. So C4 pathway can be found where? In the dry tropical regions. Okay. Friends, have you seen any C4 plants? in dry tropical pathways. You might have seen, you just don't know. Okay. C4 pathway. Now, though these plants have C4 oxaloacetic acid as the first CO2 fixation product, the first product is going to be oxaloacetate. It's 4 carbon. Okay. Students, this is the C4 plant, how they look from the inside. C4 plant looks like this from the inside. Let's take a 15 minutes break and then I'll come back and then I'll teach you C4 pathway. After 15 minutes, we will do C4 pathway and that's all. Chapter is over actually. C4 pathway and factors affecting photosynthesis after 15 minutes break. Okay. Let's take a 20 minutes break. We'll come back. I'll go drink some water and relax for some time. Let's come back and then finish C4 pathway. It's very easy. You'll get over it very fast. Okay. So it's 7:23. 7:43 we'll start again. Okay. Okay. I'll come back and I'll teach you what is this, what is the meaning of this, something called as a wreath-like structure, what is this meaning of bundle sheets, what is the inside part. We will learn all that. Okay. So I'll see you in 20 minutes. So I think we are audible, right? Let me check in the chat. If I'm audible, let me know. I think I kept the pen somewhere. 1 second, students. I think I kept the pen somewhere. 1 second. Yeah, I got it. Now, students, let's understand something called a C4 cycle. It's very simple. If you understand something called a C3, C4 becomes a cakewalk, right? C4 becomes very easy to understand if you understand C3. C4, you might think it is a little difficult to understand because there are two compartments here, right? One compartment up, one compartment below. But I will draw it and show you, and it is actually super duper easy. Very easy, very easy. Ready? Right? Ready. Okay, can we start, students? Let me know in the chat if I'm audible and visible also. Okay. Now, students, you need to understand here is, I told you here, right? I mentioned here that your Rubisco and CO2 will be locked in a separate compartment, right? It will be locked in a separate compartment where your oxygen cannot enter. Oxygen cannot enter. All of you understood? Yes, sir. Oxygen will not be able to enter there, and CO2 and your CO2 and Rubisco will be able to aram se live hereafter. Okay. Now, understand what is that room here. If you look at the diagram of a cross sectioning of a C4 plant, a C4 plant will have something called as Kranz anatomy. Now, what is this Kranz anatomy? Understand very well. Understand here. Can you see this is the upper epidermis, this is the leaf. It's a leaf section. Upper epidermis, lower epidermis. Now here, students, if you can see the vascular bundles on the inside, sorry, bundle sheet cells with large chloroplast are here. Yes. Here we have, inside we have the bundle sheets. Bundle sheets are on the inside. Bundle sheets are on the inside, and on the outside we have the mesophyll cell. Mesophyll cell on the outside. Now, students, tell me, can the oxygen enter into the bundle sheet cells? Can the oxygen enter into the bundle sheet cells? Obviously not. Your bundle sheet cells, your oxygen cannot enter into the bundle sheet cells. Oxygen might enter in the mesophyll cell. Here, it can enter here. But oxygen cannot enter the bundle sheet cells. This is what the cage I was telling. This was the room I was telling you. Within this room, within this inside room, we will have Rubisco. Rubisco will be inside. Rubisco will be on the inside here. Will be on the inside. And this is the same place where we will have CO2. Where we will have CO2. CO2 will be sent inside. Rubisco will be also be present on the inside. Rubisco will not be present on the outside. That is the mesophyll cells. So mesophyll cells will have other enzymes which I'll tell you, but Rubisco is always present in the bundle sheet. That point is clear to all of you. Tell me yes or no, first of all. So let me draw it for you. Let me draw it for you. Now, doesn't this look like a wreath? Doesn't this resemble this wreath? This is called as wreath-like structure. Wreath-like structure. So this is the wreath, where the bundle sheet cells on the inside are covered by a mesophyll cell on the outside. Mesophyll cell on the outside. Now let me draw it for you, students. Let me draw it for you. Here we have two different shapes. Here we have two different cells. Here we have two different cells. Oh, 1 second. Here we have two different cells now. Understand. Okay. Now, here we have two different cells. The above cell here. Tell me, which was the cell on the outside there? Tell me, which was the cell on the outside? The outside cell was the mesophyll cell, right? Mesophyll cell. So this right here is the mesophyll cell. Mesophyll cell. Mesophyll cell. Right? This is a mesophyll cell. And the inside cell was called as bundle sheet. It's called as bundle sheet. Bundle sheet on the inside. Bundle sheet on the inside. Now, CO2 is entering. Now here, remember students, oxygen cannot enter here. Oxygen cannot enter inside the bundle sheet. No, it is not allowed at all. It is not allowed. Oxygen has to stay outside only. Okay. In this C4 cycle, also called as hatch and slack pathway, CO2 is entering. CO2 is entering. CO2 is entering here. Okay. CO2 is entering. Now, which is the primary acceptor of CO2 here? The primary acceptor of this CO2 is nothing but PEP. Now, what is this PEP, students? PEP is nothing but phosphoenol pyruvate. Phosphoenol pyruvate. Phosphoenol pyruvate. Okay, phosphoenol pyruvate. Phosphoenol pyruvate. Phosphoenol pyruvate. When this phosphoenol pyruvate, this carbon, this is one carbon. This carbon is CO2 is going to be one carbon. This PEP is three carbon. Three carbon. Now tell me, when one carbon plus three carbon, what do we get, students? One carbon plus two carbon, what do we get? A four carbon compound. The four carbon compound is nothing but OAA, which is four carbon nature. Now, what is this OAA? OAA is nothing but oxaloacetic acid. OAA is nothing but oxaloacetic acid. Oxaloacetic acid. Oxaloacetic acid. Oxaloacetic acid. OAA. So, since the first stable product of your C4 cycle is a 4 carbon, that is why it's called as C4 cycle. Now, students, understand this OAA is now converted to an organic acid. We learned, right? OAA gets converted to an acid. This OAA gets converted to malic acid. And this malic acid is transported to bundle sheet. Malic acid is transported to bundle sheet. This malic acid, which is again a 4 carbon compound, this malic acid, which is a 4 carbon compound, gets converted back to OAA. OAA is 4 carbon. This OAA now gets converted to something called as pyruvic acid, also called as pyruvate. Okay, pyruvic acid. This pyruvic acid is 3 carbon. OA is 4 carbon. Pyruvic acid is 3 carbon. 1 carbon is missing. That 1 carbon is removed in the form of, that one missing carbon is removed in the form of CO2. Now tell me, what will happen to this CO2, students? This CO2 now enters a C3 cycle. Like how here, students, how we previously learned, this entire process will happen again. This CO2 is entering, it is taken up by RUBP. RUBP gets converted to PGA. The same thing will happen inside the bundle sheet now. This same thing, this CO2 will be taken up by RUBP, where inside the bundle sheet, which will give rise to PGA. PGA gets converted to triose phosphate. Triose phosphate again gets converted back to RUBP. This entire cycle is happening in the bundle sheet. Remember, there was a line which said C3 cycle happens in both C4 and C3 plants. This is what they meant. Even though it is C4 cycle, inside the C4 cycle, C3 cycle will take place. Inside the C4 cycle. Now, what is the fate of this RUBP, students? What is the fate of this pyruvic acid now? This pyruvic acid is sent up. This pyruvic acid again gets converted back to, where is it? This pyruvic acid again gets converted back to PEP. It again gets converted back to PEP, and in this conversion, when this conversion is happening, 2 ATP is consumed. 2 ATP is consumed to 2 ADP. So what are the cycle, students? First we have CO2, PEP, we are getting oxaloacetic acid. It's getting converted back to malic acid. Malic acid to OA, pyruvic acid. One CO2 is removed. That CO2 is taken up by your Rubisco and C3 cycle is happening. C3 cycle is happening inside the bundle sheet. Inside the bundle sheet. Pyruvic acid is again sent up to the mesophyll cell where pyruvic acid is converted back to PEP. Phosphoenol pyruvate. Now students, you tell me, do we have the enzyme Rubisco here? Yes, we have Rubisco here. Rubisco. I told you, Rubisco is trapped with CO2. Rubisco is trapped with CO2. So that is why in the case of C4 cycle, only and only there is no C2 cycle. So there is only and only C3 cycle is happening here and we are getting a product. There is no photorespiration here. There is no photorespiration here. Now here if you have Rubisco enzyme, CO2 PEP, there has to be one enzyme in this conversion. In this conversation, one enzyme should be there. The enzyme which is present here is called as pepcase. Pep carboxylase. Pepcase enzyme. Remember, they will ask you questions. They'll ask you questions. In where do you find pepcase enzyme? Remember, pepcase enzyme is present in mesophyll cell. Rubisco enzyme is present in the bundle sheet. That's how they'll ask you question. Straightforward questions they'll be asked you. Friends, if you're able to understand this, quickly smash the like button because this is a pure concept based topic. You cannot by heart this. You need to understand this. Now, if you go back here, students, if you go back here, what we were learning, we learned here, right? We learned here. I told you I will explain this later. See, read this again. Read this again. In C4 plants, photorespiration does not occur. It is because they have a mechanism that increases the concentration of CO2 at the enzyme site. So, can you understand now? At the enzyme site, CO2 concentration is increasing. Enzyme site, that is the bundle sheet, CO2 concentration is increasing right now. That's all. It's the line which they told you there. Okay, understand. Okay. Though these plants have C4 oxaloacetic acid as the first CO2 fixation product. See, the first product in a C4 cycle is OA. That is why C4 cycle. That is why C4 cycle. Right. Now, see here. That is, this takes place when C4 acid from the mesophyll cell is broken down in the bundle sheet cell to release CO2. Did we learn that? As acid is coming down, oxaloacetic is getting converted to pyruvic acid to release the CO2. Release the CO2. Yes. CO2 is being released here. CO2 is being released here. This results in the increase in the intercellular concentration of CO2. In turn, this ensures that the Rubisco functions as carboxylase, minimizing the oxygenase activity. That's all. Beautiful concept you just now learned. Every line of NCERT, you're understanding, you do not have to by heart this big paragraphs. Understand the diagram, understand the entire concept, you're good to go. Now, what is given here? C4 plants are special. They have a special type of leaf anatomy. They tolerate high temperatures. They show response to high light intensities. They can tolerate high temperatures. They can tolerate high light intensities. Right? High intensities. They lack a process called as photorespiration and have greater productivity of biomass. See here. What is given here? They have a special type of leaf anatomy done, right? A study vertical section of leaf, one of the C3 plants and the other C4 plants. Do you notice a difference? Yes, there is a difference. The particular large cell around the vascular bundles of C4 plants are called as bundle sheath cells. Do you see that? Large cells called as bundle sheath cells are, see, the middle one is called a vascular bundle. In the middle, we have the vascular bundles in the middle. Here, middle is vascular bundles. Around the vascular bundle, there are large cells. Large cells with more chloroplast. Bundle sheet. Bundle sheet. Bundle cells. And the leaves which have such anatomy is called as Kranz anatomy. The Kranz meaning wreath. And is reflected on the arrangement of cells. The bundle sheet cells may form several layers around the vascular bundles. They are characterized by having large number of chloroplasts. They are large chloroplasts, right? They thick-walled, impervious to exchange of gases, no interstellar spaces. Here, this bundle sheet cells are large, no interstellar spaces around them, and a wreath-like structure. Bundle sheet. Nothing can come inside. Bundle sheet. You may like to cut a section of C4 leaf, maize or sorghum. Examples of C4 plants. Maize or sorghum. To observe the Kranz anatomy and distribution of mesophyll cell. Mesophyll cells around it. It would be interesting for you to collect leaves of diverse species of plant around you and cut vertical sections. Observe. Okay. This is normal thing. We have learned all this now. This is called as Kranz anatomy. Now, what did we learn? We learned here, right? To study the pathway, this pathway that has been named Hatch and Slack pathway. I told you. Discovery. C4 cycle is also called as Hatch and Slack pathway. Hatch and Slack pathway is again a cyclic process. See, the primary CO2 acceptor is the 3-carbon molecule called as phosphoenol pyruvate. Yes, 3-carbon compound, that is phosphoenol pyruvate, PEP, first carbon, first acceptor. Okay, and is present in the mesophyll cell. Present in the mesophyll cell. That is very important. Present where? It present in the mesophyll cell, right? Present in the mesophyll cell. PEP, mesophyll cell. Comparison is very important. Okay. The enzyme responsible for fixation is PEP carboxylase or pepcase. And pepcase is always present in the mesophyll cell. It is important to register that the mesophyll cell lacks Rubisco enzyme. The C4 acid OA is formed in the mesophyll cell. So oxaloacetic acid is found in the mesophyll cell and it has pepcase enzyme. Rubisco enzyme is absent in the case of mesophyll cell. Here. In mesophyll cell, there is no Rubisco. Rubisco is only found in the case of bundle sheet. No. It is then the other 4-carbon compounds like malic acid or aspartic acid in the mesophyll cell leaf itself. See, they'll ask you a question. Where is malic acid synthesized? What is the answer? Tell me. Where is malic acid synthesized? Malic acid is synthesized in the mesophyll cell or bundle sheet cell. What is the answer? Now tell me. Malic acid is produced in bundle sheet cell or mesophyll cell. You are supposed to tell me, sir, malic acid is synthesized in the mesophyll cell. Then it is transported to bundle sheet. Okay. It is important to register that the mesophyll cell lacks Rubisco. See, the other 4-carbon compound like malic acid or aspartic acid in the mesophyll cell itself. See, produced in the mesophyll cell, right? Which are transported to the bundle sheet later on. In bundle sheet cells, these C4 acids are broken down to release CO2 and a 3-carbon molecule. That is pyruvate is released. Okay. Not bundle sheet. Mesophyll cell they are produced. Mesophyll cell. Synthesized in mesophyll cell, transported to bundle sheet. Diagram is very important. Understand the diagram. Write this diagram and understand. Very simple diagram if you want to follow. Synthesized in malic acid here, transported here, CO2 is released out, 3-carbon compound is produced. 3-carbon compound is produced. Here, 3-carbon molecule is produced. The 3-carbon molecule is transported back to the mesophyll cell. I told you, pyruvic acid is again sent back, where it is converted to PEP again, thus completing the cycle. So this pyruvic acid is again sent back to mesophyll cell. It is converted again back to PEP, phosphoenol pyruvate. The CO2 released in the bundle sheet cells enter the C3 or Calvin cycle. Enters the Calvin cycle again. Right. A pathway common to all plants. I told you, there's a statement before. A C3 cycle and C4 cycle. The major difference is photorespiration. But if they ask you a question, does the C3 cycle occur in C4 cycle? What should be your answer? Megara, your message is visible, don't worry. Tell me what should be your answer. Is C3 cycle present in C4 cycle? True or false? True, sir. True. C3 cycle is present in the C4 cycle. Present in the C4 cycle. Right. The bundle sheath cells are rich in enzyme rubisco. Rubisco is present in bundle sheath cell. Okay. But lacks pepcase. Thus, the basic pathway that results in the formation of sugar, the Kelvin pathway, is common in C3 and C4. It will happen. Okay. Did you notice that Kelvin pathway occurs in all the mesophyll cells of C3 plants? In C4 plants, it does not take place in the mesophyll cell, but does only in the bundle sheath cell. That is, in the case of C4 plants, C3 cycle occurs in the bundle sheath cell. That is the only thing which you need to know. C3 cycle occurs in bundle sheath cell. Beautiful. See here. Phosphoenol pyruvate, CO2. Fixation is happening. We get oxaloacetic acid. Oxaloacetic acid is converted to malic acid, transported here. Decarboxylation, CO2 is released out. Then again we have the 3-carbon acid, that is pyruvate acid. Again transferred back to your mesophyll cell. It is getting converted back to your phosphoenol pyruvate. Entire cycle is over. Clear? Such an easy thing, such an easy cycle it is. Such an easy cycle for you to understand. You don't have to by heart this. All you need to do in your C4 cycle is this diagram. Draw this diagram. You will be able to solve all the questions. There is nothing to worry about here. This is the entire C4 cycle. Okay, C4 cycle. See, this is C4. This is C2 and this is C3. Draw each one of them in every single page and compare and see. Right? Compare and see, is there any difference? Okay. Clear? C3, C2, C4, done and dusted in today's class. Don't you feel confident now? Don't you feel more confident learning this 3 different cycles now? 3 different cycles in botany. I know many students still get confused between C2, C3, C4, but if you're watching this lecture, if you're writing these notes, right, this should be a cakewalk to you. Very easy. Very easy. Now let's do a quick difference. Let's do a quick difference now. Difference. Understand the difference. Tell me if you don't understand any line here. Do you want these notes? Yes, the notes will be given in the Telegram channel, Vedantu NEET English Telegram channel, and also Baswara Sir Biology. I know I'll be uploading the notes there. Okay. Baswara Sir Biology. Don't worry. Notes will be available to you. Okay. My Telegram channel. All of you know, right? This Telegram channel, it will be available. Notes. Okay, don't worry, it will be available. It will not run away anywhere. Look at the difference and understand the concept of solidification. You will solidify the concept now. Solidify the concept. The cell type in which Kelvin cycle takes place. Tell me. We just now learnt the Kelvin cycle in the case of C3 plants in mesophyll cell. But in the case of C4 plants, Kelvin cycle is happening in the bundle sheath cell. We just now learnt. See here. C3 cycle is happening in the bundle sheet. C3 is happening in the bundle sheet. That concept, first point, is clear to everyone. Tell me. Students, I want you to respond to every single concept now. Every single line, it is clear. Thumbs up or hearts or fire in the chat. First line is clear to all of you? Yes, clear to all of you. Now second line. Cell type in which initial carboxylation occurs. Both is in the mesophyll cell. Both is in the mesophyll cell. Initial carboxylation means what? Initial carboxylation is initially CO2 is being fixed. In the case of C3 plants, it's always mesophyll. But in the case of C4 plants, initial fixation is happening in the mesophyll cell. Clear to all of you? Tell me all of you in the chat. Green has in the chat or fire or thumbs up, anything. Second point clear. Initial fixation of CO2 is happening in mesophyll cell in C4. How many cell types does the leaf fixation have? Only one in C3, but in C4 there are 2 types. Mesophyll on top, bundle sheet on the bottom. Okay, done. Okay, next one. Which is the primary CO2 acceptor? Here it is RUBP because only one. Here it is pepcase. sorry, PEP, phosphoenol pyruvate, which is receiving it. Phosphoenol pyruvate is receiving the CO2 in the mesophyll cell. Number of carbons in the primary CO2 acceptor. Here we have RUBP, 5 carbon. Here we have 3 carbon. Okay, 3 carbon. Number of CO carbon in the primary acceptor, 3 carbon here. 3 carbon. 3 carbon in the PEP. Phosphoenol pyruvate is 3 carbon. Understanding every point. Every point you're understanding now. 3 carbon. Okay, 3 carbon. Next, which is the primary CO2 fixation product is going to be PGA, that's why C3 cycle. Oxaloacetic acid, that's why it's a C4 cycle. Number of carbons, 3 carbon, 4 carbon. Does the plant have Rubisco? Yes, both of them have Rubisco. Both of them have Rubisco. All of you agree with me? Does the plant have pepcase? Here, no. C3 cycle does not have pepcase, but your C4 cycle have pepcase in the bundle sheet. Right? Does which cell in the plant have Rubisco? Here mesophyll cell have Rubisco, but in the case of C4 cycle, bundle sheet have Rubisco. Bundle sheet have Rubisco. Right here. Let me keep reminding you. This should stuck in your mind. Learn the topic today. Don't take it tomorrow. See, C3 cycle, bundle sheet. Rubisco is present where? Bundle sheet. Or CO2 is released in the bundle sheet. Bundle sheet. No. CO2 fixation rate under high light conditions will be medium. Doesn't work very well. But in high light condition, C4 plant will work very well. Because I told you, C4 plant can work efficiently in your high light and high temperature. Whether photorespiration is present at low light? No. In low light, it is not present. Here it is never present. Here it is never present in C4. Whether photorespiration is present in high temperature? Yes. In C3 cycle, photorespiration will always happen. In high light conditions, photorespiration will happen in C3 plants. It will never happen in your C4 plants. Whether photorespiration will be present at low CO2 concentration, obviously I told you in the starting, whenever there is low CO2, told you here, whenever there is low CO2, high oxygen, high temperature, high light, photorespiration will happen. CO2 cycle will always happen. Does the CO2 cycle happen in your C4 plants? No, it never happens in C4 plants. Never occurs in C4 plants. Whether photorespiration will be present at high CO2 concentration? No, obviously not. If there is high CO2, plants will always go for C3. Does not go for C2. Okay, doesn't go for C4. C2. Okay, knowledgeable. It will never happen here. Temperature, it happens at low temperature, at high temperature, I told you. Example, wheat and rice, maize, sugarcane, sorghum. Examples of your C4 plants. Entire difference done and dusted. Between C3 and C4. Clear, all of you? That is the beauty of your plant physiology. If you understand the concept, there is no way to by heart this. Once you understand the concept, you don't have to by heart any of this. Okay? Understood, all of you? Yes? Clear? Now can we go to last topic today? Can we go to last topic today? Now, last topic today. Can we start? So can you play the before slide for examples? Yeah. Don't worry, the notes will be given to you. Wheat and rice in C3. C4 is maize, sugarcane, sorghum. Clear? Now we'll come to the last topic. Now here you can expect 1 or 2 questions. Some years they have come. It's very easy to understand. So let's read one by one and let's understand. Okay, let's start. Factors affecting photosynthesis. Okay. As understanding of the factors that affected photosynthesis is necessary. The rate of photosynthesis is very important in determining the yield of the plants, including crop plants. Photosynthesis is under the influence of several factors, internal and external. Internal factors and external factors. The plant factors include the number, size and age of the orientation of the leaf. Obviously, students tell me, if there is more leaves, will the photosynthesis happen more? Yes, it will happen more. Okay, clear? If there is more leaf, more photosynthesis. If the size is bigger, leaf size is bigger, obviously more photosynthesis. So when will you start this batch? Can we join course? Shravanya, which batch talking? DNA batch? DNA batch is always there. So PYQ, these lecture sessions, we will do PYQs after the topic is over. Okay, don't worry. PYQs will happen. PYQs is there, don't worry. No, it is there. PYQ is there. Okay. Now, mesophyll cells and chloroplast and internal CO2 concentration and amount of chlorophyll. All these are internal factors. Obviously, more chlorophyll, more photosynthesis. More CO2, more photosynthesis. Right? The plants or the internal factors are dependent on the genetic predispositions and growth of the plant. Internal factors are dependent on the genetic factors. Now, what about external factors? The external factors would include availability of sunlight, first one, temperature, CO2 concentration, and water. These are 1, 2, 3, and 4. 4 different external factors. DNA batch has already started, you can still join. DNA batch has already started a long time ago, right? Now, as a plant photosynthesis, all these factors will be simultaneously affect its rate, right? Hence, though several factors interact and simultaneously affect the photosynthesis or CO2 fixation, usually one factor is the major cause or is the one that limits the rate. Hence, at a point, the rate will be determined by the factor availability at suboptimal levels. Understand this point, very simple it is. They're talking about the limiting factor. Now, predispositions means pre-made, like already it's decided. Already decided factors. Internal factors are already decided by genetic factors. Okay. Now, this paragraph is usually explaining something called as limiting factor. Understand? They're explaining something called as limiting factor. Now, you are preparing for a NEET examination. There are many factors which will be playing a role in how you score in your examination. Right? How you score well in the examination. Now, can I tell there is this one factor which is the one, there are many factors. It could be a preparation, if you're solving PYQs, if you're solving mock test, if you're, being regular and everything. Many factors are there, right? Now, can I tell, if you do not do enough of mock test, if you do not do enough of mock test, will it affect your result? Yes. Now, in your preparation, there was this one factor. Because of that one factor, if you do not do well, it is affecting your result. That one factor which is affecting your result because of the low concentration, that is called as limiting factor. For example, in the case of your plants, many factors control the bioprocess, right? Many factors control the biochemical reaction, many processes, right? In the photosynthesis, many factors control a biochemical reaction. Many factors control biochemical reaction. Then photosynthesis. Photosynthesis is rate. The photosynthesis rate is determined by that factor which is present in the least amount. Your rank, right? Your rank will be determined. Your rank will be determined by that one factor, which is you're ignoring completely. For example, if your score is low because you did not solve enough of PYQs, that is called as limiting factor. Your score is low because you did not do mock tests, that is a limiting factor. Your score is low because you did not complete the backlogs, that is the limiting factor. In the case of plants, there are many factors which includes rate of photosynthesis. Many factors are there which determine the rate of photosynthesis. But the actual rate of photosynthesis, that is your rank or the photosynthesis rate, will be determined by that factor which is present in the least amount. For example, in summertime, students, tell me, okay, let's take winter first. Let's take winter. In winter time, tell me, do you get sunlight immediately, or does it take a lot of time? Shravani, you can join tomorrow only DNA batch. There is no restriction. You can join the DNA batch tomorrow only or today only. Okay, understand. In winter time, sunlight is very low. Sunlight is very low. So can I tell sunlight is a limiting factor there? Yes, sunlight is a limiting factor because that factor which is present in the least amount is the limiting factor. Because plants require sunlight, plants require chlorophyll, plants require CO2, plants require water for the photosynthesis. If any of the factors is present in the least amount, that is called as limiting factor. It could be water, sunlight, chlorophyll, CO2 concentration, chlorophyll concentration. Now let's take summer. In the case of summer, what about in the case of summer? In summer, there is sunlight. Enough of sunlight is there in summer also, early morning. Okay, sunlight is there. But in the case of summer, students, do you have a lot of water or less water? We have a little bit of less water comparatively. Yes, in summer time there's a drought. Yes, water would be less. Water would be comparatively less. Since water is less, can we tell the rate of photosynthesis can be affected by that? Yes. So water now becomes the limiting factor. Water now becomes the limiting factor. A factor which is present in the least amount is called as limiting factor. Understood, all of you? Understood? The factor which is present in the least amount becomes the limiting factor. In summertime, it is usually sunlight. In summertime, it is water. In winter time, it is the sunlight, limiting factor. When several factors affect the biochemical process, also called as the Blackman's law of limiting factor, comes into effect. This states that the following: if a chemical process is affected by more than one factor, then its rate is determined by the factor which is the nearest to its minimal value, right? Like water was the minimum thing, water was not available, so water became a limiting factor. It is a factor which directly affects the process of quantity is changed, right? Quantity is changed. For example, in your NEET preparation, if you do not do PYQ, it will affect your rank. If you do not solve mock tests, it will affect your rank. If you do not join a test series, it will affect your rank directly. Okay? Nice. For example, despite the presence of green leaf and optimum light and CO2 condition, the plant may not photosynthesis if temperature is very low. Yes, in winter condition, temperature is very low. So temperature becomes the limiting factor there. If this leaf, if given the optimal temperature, then only it will start photosynthesis. Limiting factor. Now, for example, let's understand each factors now how they play a role. For example, light first. Light. We need to distinguish between light quality, light intensity, and duration of exposure of light. While discussing light as a factor that affects the photosynthesis, there is a linear relationship between incident light and CO2 fixation. There is a direct relationship. Now, what is the relationship between incident light? What is the relationship between incident light intensity and photosynthesis rate. What is the relationship, friends? Understand here. In the starting stage, from, let's just take this is O. From O to B, can I tell from almost O to A? First, let's take O to A. Can I tell during O to A, as the light intensity is increasing, as the light intensity is increasing, it is directly proportional to rate of photosynthesis, rate of photosynthesis. Yes, directly proportionate. As the light intensity is increasing, even the photosynthesis is increasing, directly proportionate. O to A. Okay, directly proportionate. But look at B to C. B to C. In B to C, even if light intensity increases, is there any increase in photorespiration even if the light intensity is even more increasing, is there any increase in rate of photosynthesis? No. From B to C, rate of photosynthesis is constant. Rate of photosynthesis is constant after some time. It is constant. Can you see? Even though light intensity might increase here, even though light intensity might increase even more, but the rate of photosynthesis is stagnant now. Stagnate. It's stagnate. So it tells you that in the initial stage, light can be a limiting factor. But after some time, it is not, because light is present enough everywhere. Light is present enough everywhere. See what is given here. There is a linear relationship between incident light and CO2 fixation rate at low light intensities. Can you see? Initially at low light intensity, there is a direct relationship. Direct relationship is there. At high light intensities, gradually the rate does not show further increase. That is, rate of photosynthesis is constant after some time. Here, after some time, rate is constant at high light intensities. As the high light intensity is happening, rate is constant. Okay. As other factors becomes limiting. What is interesting to note that light saturation occurs at 10% of full sunlight. So out of the sunlight, entire sunlight, only 10% of the sunlight is enough for plants. 10% of sunlight is enough for plants. They don't require all of it. Okay. Hence, except for the plants in shade or in dense forest, light is rarely a limiting factor in nature. So it does not make a limiting factor. Increase in incident light beyond a point causes breakdown of chlorophyll and a decrease in the photosynthesis. So even high light is very bad for plants. You can't increase too much and expect the plant to work really well. No. If you increase the high light conditions, plant will eventually die. Okay, eventually die. No. Next one. Carbon dioxide concentration. Carbon dioxide is a major limiting factor for photosynthesis. Yes. The concentration of CO2 is very low in the atmosphere. So can I tell if CO2 is very low? If CO2 is very low in the atmosphere, can we call this factor as a major limiting factor? Obviously. A factor which is present in the least amount is called as a limiting factor. Since the CO2 is present in a very low concentration in the atmosphere, I can call CO2 as a limiting factor, major limiting factor. Between 0.03 and 0.04%, increase in CO2 concentration up to 0.05% can cause an increase in CO2 fixation rate. So, if you increase from 0.03 to 0.05, okay, some amount of photosynthesis can happen very well. It is increasing in nature. But beyond these levels can cause damage over a long period. Does not work. The C3 and C4 plants respond differently to CO2 concentration. At low light intensities, when there is low light is very low. So obviously light is a limiting factor that time. In low light conditions, neither group respond to high CO2 conditions. Why? Tell me why. Even if there is high CO2, C3 and C4 will respond very simple. Same way. Why? Because light is a limiting factor that time. Even there is, understand students, understand. Will you get really high marks? Will you get really high marks if you only do PYQs and have a lot of backlog? No. There's a very high chance that a question might come from backlog, even if you solve PYQs. Or understand, if you're covering the entire syllabus and if you don't do PYQs, your rank will decrease again. Yes. Same thing is happening here. Even if you give high CO2 concentration, even if you give extremely high CO2 concentration, but if there is no light, plant cannot do photosynthesis. Light is very important. Okay. At high light intensities, now light is there now. Light is present here now. Light is present. Both C3 and C4 plants show increase in rate of photosynthesis because light is present now. What is important to note that the C4 plants show a saturation, that is amount of CO2 they can take is around 360 microliter per, right? Micro per liter, while C3 plants show increase up to concentration and saturation is seen only beyond 450. So can I tell C3 plants show a high saturation to CO2? C3 plants can take more CO2. If C3 plants can take more CO2, they can work very well in high CO2 concentration. Since they can take more CO2 concentration, they can work very well in high CO2 concentration. Yes. Thus, current availability of CO2 level is limited to C3 plants. Okay, limiting to C3 plants. Right. What is important to note that the C4 plants show a saturation at 360, while C3 plants respond to increased CO2 saturation and saturation only beyond C4. Thus, the current availability of CO2 level is limiting to the C3 plants. The fact that C3 plants respond to high CO2, they can respond to high CO2, right? High CO2, right? High CO2 concentration by showing increased rate of photosynthesis leading to high productivity. Obviously, if there is more CO2, right? If there is more CO2, C3 plants can work really well. See, it's given here. Leading to high productivity, has been used for some greenhouse crops. That's why, students, that's why in greenhouse gases, what happens? When the sunlight is entering inside a greenhouse, and if you enter the greenhouse, it's very hot inside because CO2 concentration is very high in the greenhouse, right? Such tomato and bell pepper, see here, CO2 concentration by showing increased rate of photosynthesis leading to the high productivity has been used for some greenhouse crops. So C3 plants can respond well in greenhouse plants. Just tomato and bell pepper, they are allowed to grow in carbon dioxide enriched atmosphere that leads to high productivity. Clear? Tomato and bell pepper can work really well in high CO2 concentration because they are C3 plants. C3 plants can take more CO2. Understand? That is the basic concept here. Okay. Now temperature. Dark reaction being enzymatic are temperature control. Tell me, students, which enzyme do you find in dark reaction? Tell me, students, which enzyme do you find in dark reaction? Rubisco enzyme. Tell me, if an enzyme can work in very high temperatures? No. Enzyme being a protein, the protein will completely denature in high temperatures. Here. Though the light reaction are also temperature sensitive, they are affected to a much less extent. The C4 plant respond to high temperatures and show high rate of photosynthesis, while C3 plant have much low level temperature optimum. All of you are aware of this. The temperature optimum for photosynthesis of different plants also depend on the habitat and they are adapted to. For example, some plants are adapted to high temperatures, they can survive. Tropical plants have a high temperature optimum than the plants adapted to temperate climates. So obviously, tropical plants can take up more heat. They can tolerate more heat. That's all. In India also you see, some people can eat more spicy food, some people cannot eat spicy food. Why? Because that is depending upon the situation where you are from. If you are from a very hot regions, for example, if you are from Rajasthan, if you are from a place like such as your North Karnataka, it's very hot there. Even in Delhi, some temperature becomes very high. So whenever there is high temperature surroundings, people there can eat high, they have high, what do I say? They can eat a lot of chilies, basically. They can eat a lot of spicy food in high temperature conditions because their body is adapted to it. Okay. Now water. Even though water is one of the main reactants, okay, students, remember water has a certain like indirect role. Water has a indirect role. Understand how? For example, students, tell me if this is a leaf right here. If this is a leaf right here, okay, this is a leaf right here. Now, what will happen if there is very less water? If there is very less water, can I tell the bulliform cells in the leaf will lose all the water and the leaf will show curling? Yes, the leaf will curve. Yes, the leaf will curve inside. If the leaf is curling in the inside, can I tell the surface area is reducing? If surface area is reducing, can I tell photosynthesis is obviously reducing because less trapping of light then. So indirect role. So decrease in water, decrease in surface area, decrease in photosynthesis then. Very simple. The second one, tell me. Second one is very simple again. If there is less water, can I tell to combat this, to combat the less water, will a stomata open or close? Think logically. If there is no water, the guard cells are flaccid. The guard cells will not open. So can I tell the guard cells will completely close? Guard cells will completely close because there is no water, right? If guard cells are closing, can I tell stomata is closing? Stomata is closed. Stomata is closed. If water is less, guard cells are closing, stomata is also closing. If stomata is closing, can the CO2 even enter then? No. So can I tell CO2 is also not entering properly? If CO2 is not entering properly, will the C3 cycle and C4 cycle happen? No. So can I tell photosynthesis is also decreased? Photosynthesis is also decreased then. That's all. That is the entire for decrease in water. It has an indirect role. Indirect role. Understand? The effect of water as a factor is more through an effect on the plant rather than directly on photosynthesis. Water stress causes stomata to close, hence reducing the CO2 availability. Besides, water stress also makes the leaf wilt, wilting of leaf, curling of leaf, thus reducing the surface area of the leaf and thus metabolic activity as well. That's all. With that, chapter is over. With that, the entire chapter is done and dusted. Now, students, are you ready to solve some questions? Tell me in the chat quickly. I will read the summary for you all once. I'll read the summary for you once. Then we will do questions. Okay? Ready? Now, this will show you that whatever you have studied right now is useful. Whatever you study right now, you can apply it somewhere now. Okay? Now, this part right now is the application part. How you can apply it, right? How you can apply it. Let me read the summary first. Green plants make their own food by photosynthesis. All of you know that, yes. During this process, carbon dioxide from the atmosphere is taken by the leaf through stomata and used for the making carbohydrates. Principally, glucose and starch is the products. Okay, products. Photosynthesis takes place only in green parts of the plant, mainly leaves. Within the leaves, the mesophyll cells have a large number of chloroplasts and responsible for the CO2 fixation. Mesophyll cells. Within the chloroplast, the membrane is site for light reaction, thylakoid membrane, while the chemiosynthetic pathway occurs in the stroma, that is the dark reaction. Photosynthesis has two main stages, light and carbon fixation. In light reaction, the energy is absorbed by the pigments present in the antenna and the funneled to the special chlorophyll, that is the reaction center. Okay, there are two types of photosystem. Photosystem 1 and 2. PS1 at 700, PS2 at 680. Okay. After absorbing the light, electrons are excited and transferred through PS1 and PS2 and finally to NAD, forming NADH, the final product. During this process, a proton gradient is created. Chemiosmotic synthesis, right? Membrane, the thylakoid. The breakdown of the proton gradient due to the movement of F0 part of the ATP synthase enzyme releases enough energy for synthesis of ATP. Chemiosmotic synthesis. Splitting of water molecule is associated with PS2, resulting in the release of O2. Proton and transfer of electrons to PS2. In carbon fixation cycle, CO2 is added to the enzyme Rubisco to a 5-carbon compound RUBP. It is converted to two molecules of 3-carbon PGA, dark cycle basically. Now this is then converted to sugar by Calvin cycle and the RUBP is regenerated. During this process, ATP and NADPH synthesized in the light reaction are utilized. Rubisco also catalyzes a wasteful oxygenic process called as photorespiration or C2 cycle. Some tropical plants show a special type of photosynthesis called as C4 pathway. In this plant's first product of your CO2 fixation takes place in the mesophyll, right? It is a 4-carbon compound, that is your oxaloacetic acid, OAA. In bundle sheet cells, the Calvin pathway is carried out the synthesis of carbohydrates. In the bundle sheet, we have the C3 cycle happening. Here is the first question. Read the question, take your time, all of you. Read the question. Read the question, all of you. Time starts now. Go. Read the question. It's a NEET 2024 question. NEET 2024 question. How many molecules of ATP and NADPH are required for every molecule of CO2 fixation? Did we learn here, students, today only? Did we learn here for every CO2 fixation? For every CO2 fixation in Kelvin cycle, right? In Kelvin cycle, we need, for every CO2 fixation, we need 3 ATP and 2 NADPH. 3 ATP and 2 NADPH, right? 3 ATP and 2 NADPH. So what is the answer now? 3 molecules of ATP and 2 molecules of NADPH is option number 3. Do you realize that? What we learned is just right here in front of you. And more than almost only 70% of students could answer this. Only 70% of students could answer this. That's all. Next question. In C3 plants, some O2 binds to Rubisco and hence CO2 fixation is reduced. Right? In C3 plants, some O2 binds with Rubisco. That is called as photorespiration. Okay. In C4 plants, mesophyll cells show a very little photorespiration, while bundle sheet cells do not show photorespiration. Students, all of you know, first statement is correct. But did I not tell you? Did I not tell you that in C4 plants, photorespiration is completely absent? Photorespiration is completely absent. Photorespiration is completely absent. So can I tell statement number 2 is wrong? Yes, very simple. They are trying to confuse you, but remember, there is no photorespiration at all in statement number 2 in your C4. So obviously statement number 2 then, yes, is false. Next question. Friends, all of these questions are from NEET 2024. This one is a difficult question. Let me explain for you. Now, what do you know? What can you tell in this? Students? In C4 pathway, it's very easy again, actually. Very easy. What happens in C4 pathway? Tell me. What happens in C4 pathway? Do we get phosphoenol pyruvate? We get enol. We get an enol, right? PEP. So A is 3. Just A is 3. Light reaction. Splitting of water. Light reaction, splitting of water. Photorespiration. Photorespiration. We get phosphoglycolate. Photorespiration, we got phosphoglycolate. Phosphoglycolate we get, right? Phosphoglycolate we get. Calvin cycle. RUBP. All of you know. Rubulose 1,1 bisphosphate. RUBP. Right? RUBP. So what is the answer? A is 3. A is going to be 3. So these two are gone. Then B is 1. This is the answer. B is the 1. C is going to be 4. Phosphoglycolate. Photorespiration. Phosphoglycolate is a product of your photosynthesis, product of your C2 cycle. All of you remember, right? Where is it? Let me show you. This is so weird. Here, phosphoglycolate. See, phosphoglycolate, C2 cycle. C2 cycle. The next question. How many molecules of ATP and NADPH are required respectively for fixation of CO2 molecule entering a Kelvin cycle? Same question repeated. Same question repeated. Okay, same question repeated. Okay, same question. All of you know ATP is 3, NADPH is 2. 3 and 2. 3 and 2. Simple. Next question. Synthesis of ATP linked to development of proton gradient across the membrane is ATP proton gradient formation. Chemiosynthetic hypothesis. Chemiosynthetic hypothesis. Very simple question. Next question. Let's see who can answer this first question. Let's see who can answer this question first now. Which of the following are required for dark reaction of photosynthesis? Let's see who can answer this question first now. I'm looking at the chat now. Do dark reaction require light? No, they don't require light. Do they require chlorophyll? No, they don't require chlorophyll. Do they require CO2? Yes. ATP? Yes. NADPH? Yes. So CDE, right? CDE. CDE. Yes. So CDE option number 2. Easy. Next question. PEP is a primary CO2 acceptor in phosphoenol pyruvate. Phosphoenol pyruvate. Not first. It is 2. Plants in dark reaction, they don't require chlorophyll. Chlorophyll is only required in light reaction. Chlorophyll is only required in the light reaction. Remember that. Okay. Phosphoenol pyruvate in your C4 plant. Right. Phosphoenol pyruvate plus CO2 in your C4 plant. In your C4 plant. All of you remember, right? Next question. It may look difficult because this question right here is actually based on two different chapters. Okay, let's see how many of you can answer this question. It's not a NEET PYQ. Deliberately I give a difficult question, so if you can understand, if you can solve this question. Friends, I'll give you a small clue. Let's see if you can solve. See, all of you know Rubisco can capture atmospheric CO2. It can capture. Okay. So A is going to be 4. So let's remove these two, this one option. A is 4. Okay. Now tell me, what does pepcase do? Pepcase. Tell me. Where do you find pepcase? In which cycle do you find pepcase? Pepcase is an enzyme you find in your C4 cycle. Pepcase is an enzyme which you find in your C4 cycle. Now see anything related to C4 cycle here? Here. Beta carboxylation of phosphoryl pyruvate. Right? So what do we get here? We get CO2 plus PEP. We get OA. C4 cycle. C4 cycle. Beta carboxylation of phosphoenol pyruvate. Carboxylation of phosphoenol pyruvate in your C4 cycle. So B is going to be 3. B is going to be 3. So can I remove this one also? Okay, I can remove this. Now, C. Cytochrome C oxidase. Cytochrome C oxidase, students. Cytochrome C oxidase is the complex. It is a complex 4. It is actually complex 4 in the electron transport system. Complex 4. Complex 4 in electron transport system. Its main work is to reduce in the last, this is the last complex, right? Before you go to ATP synthase, it reduces the water oxygen into water form. So this last cytochrome C oxidase is the last complex in the electron transport chain in which final electron is sent to the oxygen. Final electron acceptor is the oxygen, and oxygen gets converted to water. I will teach you this in the next chapter. Don't worry. In respiration plants. So sure. Now hexokinase. Where have I heard about hexokinase, students? Hexokinase. If you know glycolysis, the first step in glycolysis, glucose to glucose 6 phosphate. Glucose to glucose 6 phosphate, the enzyme there is hexokinase glycolysis. That is, catalyze the phosphorylation of glucose. So D is 2. So what is the answer now? Obviously the answer is going to be option number 3 that time. So this is a beautiful question which includes the 2 chapters. It includes both photosynthesis in higher plants as well as respiration in plants. All of you take a screenshot of this. Try solving this after the respiration in plants chapter one more time. Next question. Which of the following combinations are required for chemiosynthetic chemiosmosis? Chemiosmosis. All of you know intact membrane, proton pump, proton gradient, and ATP synthase. No. NADP synthase, no. 3 are there, but proton, this is missing here. Proton membrane is missing here. So no. This is the answer. Next question. How many ATP and NADPH are required? How many ATP and NADPH are required for 1 molecule of glucose? I've told you, glucose is C6H12O6. In one C3 cycle, we need 3 ATP and 2 NADPH. Now just multiply it into 6. Because C6, this is for 1 CO2. 6 versus 6 is 18. 18 ATP and 12 NADPH. 6 versus 12. 12 NADPH. What is the answer? NEET 2023 question. 12 NADPH. Option number 3. Yes, it will end in next 5 minutes. Don't worry. The 10 minutes will end. Next question. Following crops have been extensively cultivated in C2 rich atmosphere for higher yield. Students, the last concept we learned today, the greenhouse concept. In the greenhouse concept, did you learn that there are 2 things which grow well in the CO2 enriched environment? Which are those? That is obviously your tomato and bell pepper. We learned that. Tomato and bell pepper, right? The ratio of carbon dioxide fixation between C4 and C3. Students, this is a very tricky question. I'll give you a small clue. Compared to C3 and C4, the productivity in your C4 CO2 fixation, carbon fixation. Productivity in normal condition. CO4 carbon dioxide shows double almost. C4 plants almost show double productivity in normal condition, double productivity compared to C3. Tell me which is double here. Obviously this one, 2:1, 2 for C4, 1 for C3, double. So first one. Next question. This is a beautiful question. This is a beautiful question again. I'll read the question and understand. When one CO2 molecule is fixed, one CO2 molecule is fixed of triose phosphate molecule, one CO2 molecule is fixed as one triose phosphate, getting converted to triose phosphate. Which of the following photochemically made high energy chemical intermediates are used to reduction phase? In reduction phase, tell me, in reduction phase, in reduction phase, tell me, in reduction phase. If you go back to reduction phase, I've told you, in the reduction phase there is use of 2 ATP and 2 NADPH. Have we discussed that? Have we discussed that, students? In the reduction phase of your C3 cycle, in the reduction phase of your C3 cycle, there is always usage of 2 ATP and 2 NADPH here. 2 ATP and 2 NADPH in the reduction phase. So what is the answer? What is the answer? Tell me then. What is going to be the answer then? Oh, I should stop going back. It completely lose it. This question, yes. 2 ATP and 2 NADPH. Simple. Next question you see. The primary CO2 acceptor in C4 plant is phosphoenol pyruvate and found in mesophyll cell. Mesophyll cell is where? Top one. Okay. Mesophyll cell in C4 plant lacks Rubisco enzyme. Yes, it lacks Rubisco enzyme. Yes. Above we do not have any Rubisco enzyme. Rubisco enzyme is only present in bundle sheet. It is present only in bundle sheet, Rubisco enzyme. All of you remember that, right? Bundle sheet, Rubisco enzyme. Above a mesophyll cell, we do not have Rubisco. Next question. Which of the following is not true regarding the energy of release during ATP synthesis through chemiosmosis? Reduction of NAD to NAD plus on the stroma side, yes, happens stroma side. Breakdown of proton gradient, yes. Breakdown of electron gradient, no. This is a wrong statement. There is no electron gradient here. There is no electron gradient here. Incorrect one, incorrect. The first stable product of your CO2 fixation in sorghum. Yes. Sorghum is your C4 plants. Sorghum is a C4 plant. The first stable product is a 4 carbon compound, right? OAA. C4 plant, OAA, oxaloacetic acid. Oxaloacetic acid. The first stable product. The oxygenation of Rubisco enzyme in photorespiration leads to the formation of, I showed you the answer already, one molecule of 3 carbon compound, that is PGA. Apart from that, there is one more molecule of 2 carbon compound. Remember that, right? This is one molecule of 3 carbon, that is PGA. Apart from that, there is also one 2 carbon compound. One 2 carbon compound. Okay? Now, in Hatch and Slack pathway, the primary CO2 acceptor is. In Hatch and Slack pathway, the primary CO2 acceptor is. Again, same thing. Students, in C4 plant, primary acceptor is always and always what? PEP. PEP is taking the CO2 and it is getting converted to OA. Simple question again, simple question. With reference to factors affecting rate of photosynthesis In which of the following statement is not correct? Light saturation of CO2 fixation occurs at 10% of full sunlight, yes. Increase in CO2 concentration up to 0.5 can enhance, yes. C3 plant respond to high temperature? No, wrong statement, because we know C3 will low temperature, C4 is high temperature always. C4 can respond to high temperature, not C3. C3 is very weak. C3 cannot respond to high temperature. Incorrect statement. Incorrect statement. Next question. Phosphoenol pyruvate is the primary CO2 acceptor. Again, same question, students. Phosphoenol pyruvate, PEP, C4 plant. How many times the same question they're asked? Same question they're asked, and they're repeating it. The only question they'll ask you is primary acceptor. Where is it found? Mesophyll or bundle sheet? Such question will keep on repeating. You can answer them very easily. Okay, very easily. The process which makes a major difference in C3 and C4 is what? I told you. The major process which makes a difference between C3 and C4 is photorespiration. Photorespiration, major difference. I'll read the question if you cannot read it. A plant in your garden avoids photorespiration loss. Plant is not showing photorespiration. Has improved water efficiency, shows high rates of photosynthesis at high temperature and high temperature. Which plant is that? C4 CAM nitrogen fixer C3. Obviously, students, C4 plant. Why? Because does not have any photorespiration. Does not have any photorespiration. Has to be a C4 plant. Has to be a C4 plant. Next question. In leaves, C4 plant malic acid synthesized during CO2 fixation occurs in. Told you. Where does the malic acid is synthesized? Malic acid is synthesized where? Malic acid synthesized, is it in mesophyll cell or bundle sheet cell? Malic acid is produced in your mesophyll cell, then it is sent to bundle sheet. Then it is sent to bundle sheet. Right? Students, I don't know if I explained this enough. Many students cannot answer this question. So this is your homework question. This is your homework question. I think I explained the answer. So let's see if you can answer this in comment section today. Shravanna, Vinita, Roshini, Surya, Madhumita, I will check the comment section and I will see if you're answering this or not. Okay, in the comment section, not here. Is it bundle sheet or is it mesophyll? Where do you find malic acid? Where do you find malic acid? Okay, see here, students. I'll give you a small clue. Small clue. Okay. Small clue is, where do you find malic acid? Where do you find malic acid? Where is malic acid synthesized? Bundle sheet or is it mesophyll? Bundle sheet or mesophyll? Bundle sheet or mesophyll? The clue is here. The clue is there. Or the answer is only there, not clue. Answer only is there. Students, with that, your entire chapter of your photosynthesis higher plants is over. Okay. Students, let me tell you one more thing now. It is very important for you to know this. As I told you, limiting factor can be a very big deal. And in your preparation, there should be no limiting factor. Right. So for that, Vedantu has an amazing offer right now for all of you, which is free of cost. There's an exam called as NEET exam. If you write the exam, you can get up to 100% scholarship. You can get iPad, iPhone, mentorship class, drones. There are so many gifts. So write the exam, NEET examination. There is nothing to lose, everything to gain. Link in the description. Go register right now. Okay. Now, before I go, let me tell you, how many of you think biomolecules chapter is difficult? How many of you think biomolecules chapter is very difficult? Friends, if you think biomolecules chapter is difficult, okay. If you think biomolecules chapter is difficult, I'm going to make it easy for you. I'm going to make the biomolecules chapter very easy for you. Tomorrow, biomolecules again, line by line of NCERT at 12 o'clock. I want everyone to quickly go and smash that like button. It's tomorrow only, right? Yes, tomorrow 12 o'clock. NCERT line by line of biomolecules. How you learned plant physiology right now, how you learned the photosynthesis higher plants chapter, line by line you understood everything, you're able to solve all the questions. Same thing will happen in biomolecules if you attend tomorrow's class. And it's given right here. That is tomorrow, 12 p.m. Quickly go and mark your attendance right now. And I will see you in this class right here. tomorrow. So thank you so much, students, for today's class. Let me know in the comment section how was today's class, and I enjoyed it, right? It was really fun. It was really fun for me, right? So let's see if you're ready for this session, and I will see you in the next class. Bye bye, students. Take care, all of you. Because standing here for 3 hours and teaching the content is very difficult, right? So if you're doing it for free, I want you to let me know in the comment section, and I will see the comment section later on, and I'll see if it is worthful.